Assalamualaikum and good day everyone. So this is whereby when we don't have real time online class, okay, I'm basically going to put a link for you to view the lecture which I've pre-recorded. The notes is actually being supplied to you, and it is actually the same notes, okay, just with my voice to explain some of them. So we'll just go through some of the overview, and because it's going to be a long lecture, so usually I restrict to about 10 to 20 minutes per video. So that's why you don't panic when you see there's a lot of video to view. Okay, it's only like about 10 to 20 minutes per video, because if I were to go one whole shot, probably I will lose my voice. Right, now let's move on to the gene expression. So since this is an overview, I'm going to talk about roughly on both prokaryote and eukaryote, stressing more on prokaryote since, like I said, the first half of the course is on gene expression in prokaryote. So we know that again, genes are being expressed as a part of biological processes, whereby protein is going to be assembled from these encoding genes. So this is a necessary process in both prokaryotes and eukaryotes, and it starts with the use of information from a gene to synthesize functional protein. So what do you have as an end product from gene expression? Of course, it's protein towards the end, but you also have to bear in mind not all RNA produce protein, but definitely, okay, the end product of gene expression is functional RNA. Now, in terms of the gene expression, there are two types of gene expression: constitutive, whereby genes are constitutively expressed, and we have regulated gene expression, either the expression is being induced or repressed. And this expression, okay, whether it's constitutive or regulated, they basically are adapted based on changes in the environment. So regulation during gene expression is basically, of course, regulated during the transcription process. During the replication, it's just the unwinding of the DNA, but it's the transcription that determines whether your RNA transcript is going to be a functional RNA or not. So in prokaryote, the mechanism of regulation is usually done through operant models. In this course, we're going to cover 3 operant models, and if you remember, for some of you, if you have learned it during your bachelor degree, the 3 operant models that we usually cover or usually study is on the lag operand, error operand, or trip operand. But did you know that actually there are a whole lot more operands than those 3? Each bacteria or each prokaryote have their own operand. If you're interested, go read about it. Now, I'll just move on, okay, and refresh back your mind on the central dogma of molecular biology. So, in central dogma of molecular biology, we only have 3 components. We have DNA, we have mRNA, and we have protein. So we know that DNA replicated among themselves, okay, and DNA could be transcribed to mRNA. So mRNA or RNA, okay, so I'm just going to use sometimes RNA, sometimes mRNA, depending on the situation. So RNA on a whole generally couldn't be replicated, but in certain organisms it could. For example, viruses. And from your mRNA, it will be translated into protein. So in these processes, there are some modifications that is done along the way. Okay, those are DNA modification, which is actually not that much. But again, there's a lot of post-transcriptional modification occurring during the transcription, and this is especially whereby when your mRNA has been produced, and this is especially true in eukaryote. So you also have post-translational modification. It's actually a whole lot more as compared to this, but we're not going to cover this because our course is about gene expression. Now, first and foremost, let's have a rough outlook on the component of prokaryote and eukaryote. So prokaryote, we know that it is comprised of mostly bacteria. And eukaryote, again, it could be animal cell, it could be plant cell, it could be fungus, and so forth. So you can see the stark differences, okay, whereby you can see that prokaryote is actually very simple as compared to the complex structure, complex components of the eukaryote. Now, what I would like to highlight, okay, are the differences and also the similarities between both of them. Okay, have you guys ever seen this diagram? This is called a Venn diagram. So on one side we have prokaryote, on one side we have eukaryote. So what components do prokaryotes have? They have nucleoid and they have DNA in the form of circular DNA. How about eukaryote? It has a nucleus whereby linear DNA is located inside it, and they also have organelles. Now, what are the components that I shared between the two? Cells. Okay? When I say cells, okay, for example, okay, some of the cells within them, cell membrane, okay, the one that encloses it, of course, and ribosomes, okay, the machinery that gets the transcription and of course translation going. So more similarities or dissimilarities, okay, between them so that you can see it better. Well, you can have a better look in your notes. So I'm just showing it for your view over here. Now, in the general dogma of molecular biology, we know that these are the processes that your DNA goes through, okay, to produce mRNA via transcription and later on to produce protein via translation. So it all starts, okay, with DNA replication. So I'm actually not going to go into details. I'm going to go quite fast, okay, into this, okay. But to refresh back your mind, for some of you who have learned it, and for those who just know about this or haven't learned about this before, probably some books could help you out. So DNA could be replicated, okay, and the product is produced either in conservative, semi-conservative, or dispersive output. So the most common is, anyone can guess? It's the semi-conservative, okay, whereby the new strands contain half of the old and half of the new strand, which is quite common. When you have conservative, okay, you usually only get the one side from the parent, okay, either the father or the mother. Okay, and for the dispersive, you get bits and pieces of them being combined, okay, in one strand. Not okay. This is the differences between semi-conservative and dispersive. Okay, so one strand get from one parent for each of them, but for this one again, bits of pieces from the parents. Now, in the replication process, of course, the normal norm, like what we have now, normal norm in COVID-19, DNA can only be replicated from 5' to 3'. So regardless if you see that, okay, one is going here, one is going here, the direction is still 5 to 3, okay, it's just in the opposite direction. Don't be confused, okay? So it's called in anti-parallel. Alright, in terms of the replication, okay, to start off the replication, we have to have an origin of replication. This is the start, telling it to unwind. And replication proceeds, okay, in both directions. Like I said, in the opposite direction, you have the old and you have the new being made. So the first step, okay, is the unwinding of the DNA strand. And the enzyme involved is called helicase, just in case if you have forgotten. So helicase will bind to the lagging strand. We have two strands over here, double-stranded. One is leading, one is lagging. What are the functions? Find out. I'm not going to explain in details over here. So once DNA binds, okay, it will basically open up or unwind or unzip, okay, your double-stranded DNA. Another enzyme, okay, called DNA gyrase, okay, will be ahead, okay, either at the 5' or the 3', okay, well, depending on how you look at it. Okay, this is basically, okay, just to relieve some strain ahead of the replication fork, okay, to make sure that the unwinding and the winding back, okay, is smooth. Second step, on each DNA strand, okay, your short segment of RNA primer, okay, will be added by RNA primase. So of course it will latch onto the single-stranded DNA strand. So this is preceded by step 3, whereby nucleotides complementary to the template DNA will be added, and this is with the help of DNA polymerase, because without which it wouldn't happen. Of course, the direction is still 5' to 3', okay, in the opposite direction for both of them. So, like I said earlier on, we have two strands. One is leading, one is lagging. Leading strand is replicated continuously, which means that you will have your RNA transcript being produced, okay, in a continuous form. No chopping off, okay, no shorter pieces, smaller pieces, or not, as compared to the lagging strand. So the smaller pieces produced by the lagging strand is because it is being replicated discontinuously, okay, and this is called an Okazaki fragment. So what happens next, okay, is the Okazaki fragments, okay, basically are joined by the, well, you should have guessed it, DNA ligase, okay, the only enzyme that joins. So this is the overall process of what I've explained in the previous slides. Okay, you can look at it, you can ponder on it, you can, well, study in details if you want. It's all in your notes. Right, so this is what happens, okay, in general. Okay, parental molecule, okay, it will be unwind. So one from the original strand, one is going to be leading, one is going to be lagging. Which one is which in this, okay, don't ask me. So they will basically, okay, have a complementary strands being added to them. So in the end, they will have, okay, two double strand strands as compared to only one strand in the beginning. Now, those are the replication. So I'm going to go ahead, okay, with the transcription. So replication and transcription, just going to go ahead in one video, and then probably, okay, I'll move on to translation, probably not too detailed. Alright, transcription, which is the heart or the core of this course. So the process is more or less similar to DNA replication, but of course it is said to be less accurate. Okay, the error rate is about 10 to the power of minus 4. Why? Because of some mistakes or some problems that could occur, which will be highlighted in a while. So RNA polymerase, again, is the one that will catalyze, okay, the transcription, is the important enzyme. So synthesized RNA is not going to be base paired to template DNA. It is going to be on its own, okay, not like your replication process. So only certain parts of the genomes are going to be selectively copied depending on where the start codon is going to be. So there are 3 steps over here. So the steps are actually quite similar to the replication. It's just that I am going to classify them. The first step is going to be initiation. So it starts with a sigma factor that needs to bind, okay, it is a tough protein that needs to bind to your RNA polymerase to form holoenzyme. So when I talk about, or when I say holoenzyme, okay, what immediately should come to your mind is the basically complex of RNA polymerase plus sigma factor. So this holoenzyme will bind tightly to the promoter region on your DNA template or double-stranded DNA. Transcription can only start or initiated when the structure of the promoter holoenzyme complex unwinds DNA to form the transcription bubble. So transcription, okay, starts with the unwinding, similarly, almost similar to the replication. And okay, we call this as transcription bubble. So transcription bubble is going to be a bit bigger as compared to the unwinding in the replication process. And of course, RNA is then going to be synthesized still in the 5 to 3' direction. And okay, basically, okay, it's going to start at around 10 nucleotides from the promoter, not exactly at the start of the promoter, okay, about a few nucleotides away, about 10 nucleotides away from the promoter. So this is what happens, okay, whereby, okay, when you have the RNA polymerase, okay, plus the, well, the whole sigma factor is not here, okay, so when it binds, okay, it will form a closed complex. So when the transcription starts, okay, it will form an open complex, and of course, okay, initial transcription could occur to produce your RNA transcript. So step 2 is the elongation, and not to scare you, but elongation in eukaryote, it has 2 or 3 steps. Alright, so after about 10 nucleotide, sigma factor is going to be released. But when you see a lot of the diagrams, again, in books or over the internet in Googles, you'll see that sigma factor is sometimes being released towards the end. Actually, it's not true. Okay, sigma factor is actually being released quite early on, okay, in the process. So either way, okay, don't worry about that. So once sigma factor is being released, holoenzyme, okay, it's still called holoenzyme, okay, even though only RNA polymerase is there. It moves along the template strand, adding the RNA nucleotide at a time to the growing RNA transcript, okay, depending on the complementary sequences. So as it is being synthesized, template DNA in front is unwind, okay, for elongation, and the ones that is being left behind is reannealed to close it back. Okay, so the bubble size, okay, or the transcription bubble remain constant throughout as it moves along the strand. So RNA polymerase at the same time, okay, when RNA transcript is being produced, also proofread to ensure correct sequence is being produced. This is done by pyrophosphorolytic, pyrophosphorolytic, okay, which is removal of free incorporation, because sometimes RNA is being added along the way, and also hydrolytic, removal by cleaving. Okay, so proofreading is done a lot, especially in eukaryote. So the third step is termination. So simple, only 3 steps, right? So after sufficient length of RNA transcripts, terminators, okay, in the form of DNA sequences, will terminate transcription to release your RNA. And there are 2 types of terminator. One is called row-dependent, which requires the row protein. The other doesn't require row protein. It's called row-independent, but it requires 2 sequence elements. Okay, so one is a short inverted repeats, okay, about 20 nucleotide size, and one is about 880 base pair. However, you have to bear in mind that RNA polymerase could sometimes become arrested under certain conditions. What I meant by arrested is that they couldn't move forward. Of course, it shouldn't move backwards, but sometimes, okay, it moves in reverse, it could also occur. So what happens is that if it couldn't move forward or it moves backward, it will damage the DNA strand, and you will not have any transcript, right? So no transcription could occur. So in this case, okay, TRCF will come to the rescue. TRCF is transcription repair coupling factor protein, which will remove and repair RNA from arrested RNA polymerase. It's actually a complex process, so I'm not going to cover it. Now, okay, this is from the elongation up until the termination overall. So I'm going to show you, okay, in this diagram. So this is the overall diagram. So I think I'm going to go into it bit by bit. So at the first, okay, the start, whereby, okay, holoenzyme needs to be formed. Okay, so that means that sigma factor will be associated with RNA polymerase. Okay, so it will then, okay, this holoenzyme will bind tightly to the promoter. So it will create closed complex. Okay, so it binds closed complex. What happens next, okay, is that, okay, it will bind tightly to promoter to unwind double-stranded DNA. So once it unwinds, this is what we call as open complex. So RNA transcript is going to be produced, okay, with the help of RNA polymerase, and of course DNA polymerase, okay, that will add complementary sequence. So nucleotide, okay, NTP is nucleotide, okay. Complementary to DNA at the start site is the first RNA nucleotide. So 2 phosphate groups, okay, in the process, okay, are cleaved from each subsequent nucleotide to form RNA nucleotides. So this is of course, okay, added to the 3' end of the growing RNA transcript. Okay, so 3' end. So this is 5', 3', so it is added bit by bit like this. Okay, as you can see, this is what I meant, okay, by this statement. Yeah, so the phosphate group is being cleaved, because without which then it couldn't be added. The next strand or the next nucleotide couldn't be added. So once enough transcript have been produced, or basically a sufficient length has been achieved, okay, basically it's going to be terminated. And this is what I meant just now early on when I said sigma factor is released as RNA polymerase moves beyond promoter and along the DNA template. This looks like it's going to be released towards the end, but actually it was released quite early on. Alright, so termination. So termination either by row dependent or row independent. In this case, okay, in the overall picture just now you see that they are side by side, okay, but I'm going to show you first on the row dependent, whereby you need to have a row protein over here, row termination protein. It will basically bind, okay, to your RNA transcript. So what happens is that, okay, it will move, okay, along the RNA. So when it moves along the RNA, what happens is that, okay, it basically couldn't, or not allowing new nucleotide to be added. So when no nucleotide could be added, okay, it stops the process or it terminates the process. As simple as that. So RNA is then, your RNA transcript is going to be released, and your row termination protein will also be released, and you get your mRNA. So as compared to when you have termination by row independent terminator, you don't have the row protein. What you have are 2 sequences. Remember just now, one is the 20 nucleotide inverted repeats. One is the 880 sequences. So what happens is that, okay, it will form a GC-rich stem loop, or it will form a loop, okay, because, okay, it couldn't, basically it will bind to another. Okay, so what happens is that no transcription could occur because it has formed a loop. Okay, it couldn't move along. So it tells it to stop, and just like that, okay, transcription stops, terminated, the RNA transcript is going to be released. I'm just saying that it's going to be released, but there are sequences of the process that occurs. Right? So there you have it. This is the process of replication and transcription in prokaryote. So This is what we are going to cover in the first video. So in the next video, we're going to take a look at the translation. Okay, so let's move on to the next video.